It's my pleasure to introduce today's speaker. This is Alex Haafke. She is the master's student in my lab, and we actually had the honor of having her as an undergraduate as well. And I usually try to scare the undergrads away by having them work with Yuba, but she managed to come back and for our second round, and she's been working on her master's degree. And so today's presentation is summarizing what she's been studying the last couple of years. Take it away, Alex.
Thank you. So I would like to thank you guys for coming and coming to my exit seminar that I'm going to talk about my research that I've done over the past couple of years, which has been over the management of Diaporthe-infected soybean seed. So Phomopsis seed decay, or Diaporthe seed decay, is caused by Diaporthe longicula and other Diaporthe species. And the reason why I have both names there is because due to the one fungus, one name movement, Phomopsis is kind of the old nomenclature at this point because this is the anamorph of this fungus. But now it's commonly referred to as Diaporthe instead. So it's been suggested recently to change the name from Phomopsis seed decay to Diaporthe seed decay. Some symptoms you might see out in a field when you go looking for Phomopsis seed decay look similar to this picture here. And when you harvest the seed, you can see you might have smaller grain, more oblong seeds, and they might be wrinkled and then have a white and chalky appearance. And that white chalky appearance is actually the growth of the fungus on the seed. But it's not always this obviously symptomatic. You can have infected or Diaporthe-infected seed and the symptoms aren't very obvious and the seed doesn't look that different from healthy seed. And overall, this leads to lower germination and lower stand in a field, and it can lead to yield loss after harvest as well as economic losses. Next, I'll talk about the disease triangle. For any disease to develop, you need a virulent pathogen, a susceptible host, and a conducive environment. And for Diaporthe seed decay, or Phomopsis seed decay, the virulent pathogen is Diaporthe. It's soil-borne, seed-borne, or can reside in crop residue, and it's endemic in all of the soybean growing regions in the United States. The susceptible host in this case is soybean, and there are varieties that have some resistance to Phomopsis seed decay, but this is not a trait that is commonly selected for by breeders. And lastly, you need the conducive environment for this disease to develop. And the conducive environment is high relative humidity and high amounts of rainfall, as well as temperatures between 68 and 77 degrees Fahrenheit. And when this conducive environment happens after flowering, you can have increased chances of having Phomopsis seed decay, but it's especially important for the disease development when this happens at R7 to R8, or closer to harvest for more than 2 consecutive days. And back in 2018, we saw this very conducive environment for Phomopsis seed decay to develop. This graph shows the average rainfall in September from 2015 to 2020, and you can see that in 2018, the average rainfall that month was about 8 inches, which is a lot higher than the rest of the years in September. And that blue line that's going across the graph is the 30-year average in September for Iowa. And that's at about 3.5 inches. So this was a pretty extreme weather event that happened in 2018. So after all of this rainfall, the fields that were harvested were subjected to increased moisture of the pods and the seeds, which led to an increase in Phomopsis seed decay. Next, I'll show a graph that shows the estimated yield loss due to Phomopsis seed decay in the United States and Ontario, Canada from 2015 to 2019. And from 2015 to 2017, the amounts of bushels lost due to Phomopsis seed decay were pretty low, but there is a huge spike in 2018, and this can be directly linked to that weather that happened in September. And it was estimated that there was 87 million bushels lost, which was the equivalent to over $1 billion lost. And when farmers went out to harvest that fall, the seed that they found was fairly alarming. there was visible mold growth on the seed, and this led farmers and seed companies to ask, what do we do with this seed? So seed companies will send their seed for germination and seed health testing. Two common tests is a standard worm germination test as well as a blotter test. For the worm germination test, this is done in 25 degrees Celsius for a week on cellulose crepe paper. And then afterwards, you can get an estimate for germination under ideal conditions, as well as seeing if there's any abnormalities in your seedlings. And next, for blotter testing, this is really helpful when you have seedlots that have high fungal pathogen or storage fungi present, because as you can see, there's fungi will look morphologically different in this test. And again, after about a week of incubation, you can read these, and circled here in blue is what Phomopsis seed decay looks like on the seed. But there were other pathogens and storage fungi present in this seed lot. So we wanted to find seed for my research that was high or low germination with high colonization of Phomopsis seed decay or Diaporthe, which we would consider the bad seed lot. And we also wanted to find a high germination seed lot with low colonization of Diaporthe, and we consider this to be the good seed lot. So throughout the rest of my presentation, I will refer to the seed lots as being either good or bad. As I mentioned, the weather conditions at harvest in 2018 were conducive for Phomopsis seed decay development. And we collected seed that was harvested before all of that rainfall, which was what we consider to be the good seed lot. And we collected seed that was collected after all of the rainfall, which ended up being the bad. And you can see in this graph, the blue bar shows the dark blue bar shows the bad germination seed, which was only at 13%. And the good seed lot had an estimate of 99% germination. And for infection of Diaporthe, the bad seed lots had 27% Diaporthe infection, and only 2% was seen in the good. And again, these seed lots were harvested at different times, but they were the same variety. In 2019, the field conditions at harvest were much better, so the disease development of Phomopsis seed decay was really low, and it was harder to find what we consider a bad seed lot to plant for 2020. But we did find a seed lot that had 86% germination and for the bad, and then a good seed lot that had 96% germination. And then for the bad seed lot, the infection was at 16%, and the good seed lot had 8%. I wanted to see what species of Diaporthe was actually on the bulk seed that we were planting each year. So after I did the blotter testing, I took seeds that I suspected to be or to have Diaporthe, and I plated it on SAC PDA first to minimize any bacterial contamination, and then plated it on PDA and made transfers to get a pure isolate. And after I got a pure isolate, I did DNA extraction and PCR of the ITS region and did gel electrophoresis to determine which samples were considered to be positive Diaporthe isolates. And then we sent the purified PCR product for sequencing, and we found that Diaporthe longicula or Diaporthe calabra were the species that were found on the bulk seed in both years. And you can see that morphologically on the plates, they kind of look a little bit different, but when you're looking at the seed, there's no way to really tell what species is present. So using the seed lots that we collected in 2018 and 2019, we designed 3 field experiments to test in 2019 and 2020. The first experiment was a seed treatment experiment. The second experiment was a seeding rate experiment. And the third was a yield potential experiment. And first, I'll talk about the first objective, which was to test the response of seed treatments on the high and low germination seed lots. The data that we collected each year were the same for all experiments in all locations. And we collected stand counts multiple times throughout the growing season. we collected plant greenness using a green seeker that measures NDVI, and this was an average of the entire row. And we also collected canopy coverage data using the Canopio app. And this is an application that you download on your phone, and then you can take a picture of a certain area of a row, and it will convert it to a % canopy coverage. And lastly, we harvested the 2 middle rows for our yield data. And before I get into any of the results, I wanted to talk about what growth stages we collected this data at, just because, just as a refresher for everybody. We collected data at VE, which is at emergence, at V1, which is when there is the first trifoliate, at V3 is when there is 3 trifoliates, at V6, that's not shown in this picture, there's 6 trifoliates, and then at R1, which is the beginning of flowering for soybeans, and at R2, which is when flowering is complete. So for the seed treatment experiment, it was conducted over 2 years. There were 2 locations that were both in central Iowa each year, and there were the 2 seed lots of the same variety planted each year. These plots were planted at 125,000 seeds per acre and a randomized complete block design. And we tested 5 seed treatments. The first one was an untreated control. The second one was a base plus insecticide, and this is an industry standard. It has multiple modes of action and an insecticide, so this is a common seed treatment that you would see any farmer using on their seed. And next we wanted to kind of break this down and test individual modes of action as seed treatments. So we tested an SDHI, which is fluoropyrim for our study. QI, the QI that we studied was pyraclostrobin, and the last one was a DMI, which we used ibuconazole for. Next I'll talk about the stand count, or the results of the seed treatment experiment. First I'll talk about the stand counts at V3 only, just because the stand counts followed a similar trend at VE and V1 for each year at both growth stages, or at all of the growth stages. So in 2019 and in 2020, we see that seed lot had an effect on stand count, where the good seed lot had significantly greater stand. In 2019, the good seed lot had about 28% greater stand, while in 2020, the good seed lot only had about 11% greater stand. And seed treatment had an effect on stand counts in both years, where the base treated seed, which is the dark gray bar, had the highest stand counts in each year. Moving on to plant greenness, which was done using NDVI. This is a value that is given from 0 to 1, 0 being no greenness and 1 being the greenest that you could have. So at V3 and at R2, there was a significant difference based on seed lot, where the good seed lot had significantly greater plant greenness at both growth stages. And for seed treatment, we see that it only had an effect on plant greenness at the first growth stage that we took this data at, where again the base treated seed, which is that dark gray bar, had the greatest plant greenness. Moving on to the plant greenness in 2020, we see that, again, similar to 2019, there was a significant difference in seed lot, or significant difference in NDVI based on seed lot for both growth stages, where the good seed lot had significantly higher higher NDVI. And this year, we didn't see that seed treatment had an effect on plant greenness at all. For the first growth stage, the NDVI was at about 0.45, and at R1, it was from 0.8 to 0.9. So moving on to canopy coverage, again, this was out of a percent from 0 to 100, 0 being no canopy coverage and 100 being full canopy coverage. And we see that at V6 there was a significant difference where the good seed lot had higher canopy coverage, but at R2 it wasn't significant, although the good seed lot did have numerically greater canopy coverage. We saw that seed treatment had an effect on the canopy coverage in V6 and at R2. And in both years, or at both growth stages, the base treated seed resulted in the highest canopy coverage. And moving on to the canopy coverage in 2020, we see that at R1, there was a significant difference where the good seed lot had significantly higher canopy coverage, but at R2, it was no longer significant, but the bad seed lot actually had numerically higher canopy coverage than the good. And in this year, seed treatment did not have an effect on the canopy coverage at either growth stage that we took the measurement at. The last results that I'll talk to you about are the yield results from both years. So looking at seed lot, in 2019 there was a significant difference in yield based on seed lot, where the good seed lot had about 9% greater yield than the bad. But in 2020 there was no significant differences between the seed lots. And for seed treatment, we didn't see that seed treatment had any effect on yield in either year. But in 2019, the seed that was treated with the base seed treatment, which is the dark gray bar, had the numerically highest yield, and then in 2020, the seed treated with iconazole had the numerically greatest yield. So in summary, we saw that the good seed lot had greater stand and plant greenness than the bad seed lot in both years. In 2019 and 2020, the good seed lot had significantly greater coverage only at the earlier measurement. And in 2019, the good seed lot had about 9% greater yield than the bad seed lot, but in 2020, we did not see that seed lot had an effect on yield. In both years, the stand counts at B3 was greatest in the base treated seed. Seed treatment did not consistently result in greater plant greenness, and the base treated seed had the greatest canopy coverage only in 2019 at both of the growth stages, and seed treatment did not have an effect on yield in either year. And lastly, no interaction between the seed lot and seed treatment suggests that both seed lots responded similarly to the seed treatments. Now that I've covered the seed treatment experiment, I'm going to move on to the second experiment, which was the seeding rate experiment. And the second objective for this experiment was to determine if planting at a higher seeding rate can offset low germination seed. So for this experiment, again, it was conducted over 2 years. There was one location, both in central Iowa each year, and 2 seed lots of the same variety were planted in each year. The plots were planted at 4 different seeding rates, ranging from 125 to 245,000 plants per acre, or seeds per acre. And again, it was planted in a randomized complete block design. Looking at the stand counts based on seed lot, we see that in 2019, at VE and V1, it was not significantly different, but the good seed lot did have numerically higher stand counts at both of those growth stages. And at V3, there was a significant difference where the good seed lot had about 25% greater stand than the good. In 2020, seed lot had an effect on stand counts at every growth stage, and the good seed lot had about 10% greater stand at all of the counts. And I'm not going to show you the effect of seeding rate, but it did follow the trend where the lowest seeding rate resulted in the lowest plant population, and the greatest seeding rate resulted in the greatest plant population. And that was true for all of the growth stages in both years. Next, I'll talk about the plant greenness in 2019. We see that seed lot does not have an effect on plant greenness in 2019 at V3 or at R2, but seeding rate does have an effect on plant greenness. So at plots that were planted at 205 and 245,000 plants per acre, we saw the greatest NDVI, or plant greenness, in both of the growth stages. In 2020, the plant greenness, seed lot had an effect at the first growth stage where the good seed lot had significantly higher plant greenness than the bad, but at R1, there was no longer a significant difference. And again, we see that seeding rate did have an effect on plant greenness in 2020. There At both growth stages, the 3 greatest seeding rates, which were 165, 205, and 245,000 seeds per acre, also resulted in the greatest plant greenness. Moving on to canopy coverage, we see that in 2019 there was not a significant difference based on seed lot for either growth stage. And when we look at seeding rate does have an effect on canopy coverage, where the 2 greatest plant populations, which are the light gray and dark blue bar, also resulted in the greatest canopy coverage. In 2020, the canopy coverage was not significant at the first growth stage, but it was significant at the second, where the good seed lot had significantly greater canopy coverage than the bad. And we see that seeding rate had an effect on canopy coverage as well, and this was at both growth stages. So at R1, the lowest seeding rate had significantly less canopy coverage than all 3 of the other seeding rates, but at R2, the 2 highest seeding rates resulted in the greatest canopy coverage. And lastly, I'll talk about the yield for the seed rate experiment. When looking at seed lot, we see that in both of the years, seed lot did not have an effect on the yield, but the good seed lot did have numerically higher yield in both years. And lastly, when looking at seeding rate, in both of the years, seeding rate did not have an effect on yield. So to summarize the results, we see that the good seed lot had a greater plant population only at B3 in 2019 and at every count in 2020. Seed lot did not have an effect on plant greenness or canopy coverage in 2019, and it was not consistent at both growth stages in 2020. And although the bad seed lot generally had lower stand counts, this did not affect yield. As I mentioned earlier, plant population followed the trend of increasing seeding rate, where the lowest seeding rate resulted in the lowest plant population and the greatest seeding rate resulted in the greatest plant population. The 2 highest plant populations had the greatest plant greenness and canopy coverage in both years. And lastly, yield was not affected by seeding rate. So now that I've finished talking about the second experiment, which was the seeding rate experiment, I'm going to move on to the yield potential experiment. And this was to test the third objective, which was to determine if low germination seed affects yield potential when compared to high germination seed at the same plant populations. Again, this experiment was planted in 2 years. There were 2 locations each year, both in central Iowa, and then 2 seedlots of the same variety were planted each year. And the setup of planting and stand counts for this experiment was a little bit different than the last ones, because these plots were planted at a little bit higher seeding rate to make sure that we had over 100,000 plants per acre. And then we went out and we took stand counts, and using the clipper shown in this picture, we thinned the rows uniformly to achieve approximately 100,000 plants per acre. And then we went out 2 weeks later again to do additional thinning, to do another stand count and additional thinning if we needed to. For these results, I'll only talk about the main effect of seed lot in both of the years. So in 2019, we see that plant greenness was affected by seed lot, but the good seed lot had significantly less plant greenness than the bad. But in 2020, we don't see that seed lot had any effect on plant greenness. For canopy coverage in both years, seed lot did not have an effect, and they were about the same in 2019, and the good seed lot had a little bit higher canopy coverage. The bad seed lot had a little bit higher canopy coverage than the good. And lastly, when we looked at yield, we see that in both of the years, yield was not affected by seed lot, and in both of the years, the bad seed lot had a little bit higher yield than the good. So in summary, we saw that plant greenness was different in seed lots in 2019, but not in 2020. Canopy coverage and yield was not significantly different between the seed lots in either year. And lastly, low germination seed did not affect yield potential. So back to that original question of what do we do with the seed? And my recommendation is don't panic. Based on our experiments under the field conditions that we had, we saw that seed treatment increased field emergence for both low and high quality seed. Plots with plant populations above 90,000 plants per acre resulted in the same yield regardless of seeding rate or seed quality. And lastly, low quality seed affects germination and stand, but once stand is established, there were no further issues. So I would like to thank you all for coming to my exit seminar. A special thank you to my advisor, Dr. Gary Mueller, and my two committee members, Dr. Mark Licht and Dr. Gary Munfeld. And a huge thank you to the Mueller Lab, especially Yuva. He's taken a lot of phone calls about stats for me over the past few months. And I would like to entertain any questions if you have them.
What I understood from what you were saying, Alex, was that you used that 2018, the seed lot grown in 2018 and 2019, the seed lot grown in 2019 and 2020, right? That was consistent. Same two seed lots used in all these experiments, right?
So, no. The seed that was collected in 2019 was planted in the seed that was collected in 2018 was planted in 2019. And then we collected completely different seed lots in 2019 to plant in 2020.
Yeah,
does that answer your question?
No, that's just what I said.
Okay.
So I just want to make sure that I got the right impression. Yeah, okay. And so you had two low quality seed lots, one in 18 and one in 19,
yeah.
And you had your high quality seed lots. Could there be an interaction between year and seed lot, because you weren't obviously planting the same seed lots consecutively in 2 years? You had two seed lots in 1 year, two seed lots in another year. Is that something that you thought about? Or?
We did not. If it was not clear, the analysis was separate by year. So the years were not combined because the seed lots were different. So we did not combine across years.
Yeah. Okay. So getting back to your question, our growers were asking questions about what to do with these lots. So you would say to them what? Use the fungicides?
I would say that, after you sort out the visibly symptomatic seed, you can treat it with a seed treatment. You can make sure that the plant stand is kind of above that 90,000 threshold. And then after that, once you have stand that is established, it doesn't seem, at least under our field conditions, didn't seem to be any further issues after that. So.
Okay, final issue. Then in a seed treatment plant, a lot of those real bad seeds would be eliminated, right? So, right? And obviously your bad lot, you didn't run through a seed treatment plant, right? Your worst bad lot.
The worst bad lot did have all of this, visibly symptomatic seeds. Those were separated out of it. Those were separated out.
Oh.
Yeah.
Okay.
The bad seed lot we got after it was cleaned.
Yeah.
And it was still at that low of a germination.
Wow.
So I had a question about the yield loss data. So not your research actually, but you showed some data from the Final Progress publication.
Yeah.
So, the thing about phomopsis that I've always felt like the damage is done in next year's crop. So I'm a little curious. So with the high yield loss in 2018,
right.
These were beans that just were not harvestable because they were so decayed. Is that why? Is that what is behind that yield loss number?
I think that this is after it was harvested, and then some of it that clearly is visibly moldy and not usable, and then also the amount lost is due to actually selling the seed, and you can't there's discounted prices when you have that low quality seed.
This would have been, a number that big. It's not.
It's
not seed. It's not for seeds.
For
seed
It's for bushels,
Yeah.
but this is across like 29 USD for reference, because I think if you don't know that it's like all of the basically all of the soybean growing states, that's why that number is so big.
Yeah.
So if you just looked at Iowa, it's not as much as that.
Yeah, I understand. So it's either discounted prices or discarded product,
Right.
or just low yield because you have shriveled up.
Yeah, smaller states too.
So is the germination test for seed lots not that accurate? Or overestimating the
So-
germination? Because you didn't have that big a differences in stand counts, but the germination test looked like it should be
Right.
like 80%.
So there's kind of two things that play into that. One, yes, there can be kind of this discrepancy based on germination testing and what happens in the field, because what's in the lab can be way different than what your field conditions are. But the second point to that is there have been studies that show that Diaporthe can kind of die off during storage. So that could have been one of the things that we were seeing, is that seed lot, it was really bad at that point when it was tested, but then after storage and maybe some of that Diaporthe died off, and so it wasn't so hard on the seed.
And so was it, did you test germination more than once, or just?
I didn't test germination more than once. I did do water testing more than once.
Okay, and did you see that it went down over time?
Yeah, it went down a little bit when it was stored. When it was stored in the fridge, it didn't go down as much. When the seed was stored in a shed, it went down a lot, which is kind of funny. But so yeah, I did see that it kind of like went down a little bit. It wasn't a ton, at least not like in my test, but that could be different based on other people's testing.
So I think the worm germination test can be kind of erroneous when you have a lot of fungal contamination, because it's supposed to be ideal conditions for the soybeans, but it's also ideal for the fungi. So you have a lot of cross contamination and on the Kimpack, and so I would say that's probably why it performed better than the germination data suggested that it would.
Yeah.
Sometimes people do these cold germination tests. You didn't do that, right?
No, I didn't do any.
But that's supposed to be simulating a stressful spring where the soil is cold and wet, right?
That would be, that's like more of a vigor test. So we didn't do vigor testing on our seed, but that would be if it was like more of a stressful spring, or if it was planted a little bit earlier or something, and we didn't do that test. But I think that generally for seed with Phomopsis seed decay, when you do those, like that cold germination test, it's covered in sand, and the sand can kind of like help pull the coat off of the seedling, and the seed, like those counts can actually be kind of overestimated as well, which is interesting. Steve?
Does the fungus overwinter in the soil? So in 2015, 16, 17, was it there? Conditions tests weren't right?
Right,
does it sort of roll in?
It's endemic, so I think that just in general, it's always there. But then when you have those conducive environments, that's when you see the effects of it. So I would say that it is always there. It's always in the soil. It's just whether or not the conditions are right is when you get the disease.
Does it have any other hosts, or is it just that?
Diaporthe is, you can find it on weeds and stuff too. There are other plants that it affects as well. That's not just like soybeans, like other, like completely different crops. But yeah, so it can be found in weeds, but I think, I'm not sure, I have some literature on this, so I'd have to go and look it up, but I'm not sure if those species are also disease causing on soybeans or not.
If you take a bad lot, really bad lot, and you plant it, and the emergence is not good, is it possible to determine whether the Diaporthe was responsible? the pathogenic aspect of the Diaporthe in the spring was responsible for the poor emergence? Or was it just weak seed that was attacked by other things? I'm just trying to figure out when. I would have thought that Diaporthe colonized the seed from the pod, so later in the season. So I just wonder what's killing that weakened seed earlier.
What's killing the seed,
Is
like
it
right after it's
Diaporthe?
planted?
Could Diaporthe,
Yeah, I think there can be damping off due to Diaporthe if the infection is bad enough. I think that generally, like maybe if it's kind of localized to the seed coat, there might not be that, like the seed won't actually be dead, but the Diaporthe can kill the seed kind of right off the start. So.
And Mark, the way I would say it is like sort of like a race.
Yeah.
If the conditions are better for the fungus, then it has a better chance of colonizing that little seed or seedling.
somebody did a PhD on this back in the 80s. But precisely that.
Yeah. Back here.
I had it.
Lost my train of thought. So, regarding the 87 million bushels that were lost in 2018,
Yeah.
say a farmer takes a truckload of soybeans to the elevator and you lose those beans. The shriveled beans, did you do any testing with like the test weights between healthy and shriveled?
No, I didn't.
Because I'm curious if it's like, and this is kind of regarding the shriveled beans from fungus, more or less, not just ponces, but is it a density issue or is it a size and development issue?
I mean, I think that like it can actually be smaller, so I don't know if like it could be a combination of things maybe there. So like actually physically smaller size, and then also having the wrinkled, maybe like less seed moisture could impact the weight of it too.
Right. Yeah, question?
No, I'm good.
You're good. Any final questions? All right,